Okay, here we have section 18.3 talking about bacteria and the genetics of bacteria. And just real quick, the reason in chapter 18 here that we're looking at viruses and bacteria and their genetics and their life cycles and things like that is that the studying of viruses and bacteria, because they are relatively simple compared to eukaryotes, a lot of studies have been done on bacteria and viruses, and a ton has been learned about how DNA works and how genetics works. And so if we can understand some of the mechanisms of things that happen in bacteria, we're going to be able to understand some really important research that has been done and that is being done. So that's why we're kind of focusing in on viruses and bacteria here. But real quick on bacteria, what we're going to try to do is look at if I have a bacteria, a certain bacteria, I'm going to draw a bacteria here, how do we get different types of bacteria from that one? So here's a bacterial cell. If you remember, they don't have a nucleus at all, they just have a circular chromosome. I'm going to draw it like this. There's our circular chromosome. You wouldn't be able to look at a bacterial cell and see a circle like that. You would see a dense region of DNA, the nucleosome, I think, or anyway. So there's our circular nucleoid. That's the word I was looking for. There's our circular chromosome. If you remember, binary fission is the way that we're going to make a new cell like this. And basically with binary fission, this bacterial cell is going to make a copy of its DNA. So we'll make another copy of the DNA. Kind of looks like eyeballs there. Copy of the DNA. So now we have two circular chromosomes, and then this thing's going to split in half. And I'll end up with two cells, each of them with a circular chromosome. The way that we made a new copy of the circular chromosome back here was just the process of DNA replication. It's just with the circular chromosome. So these two chromosomes, and these, should be genetically identical, with the exception of the rare chances that there might be a mutation where an A or a T or a C or a G was messed up. Okay, so that would be relatively rare. So one of the things we're looking at in this section is how do bacteria, if they're supposed to be clones of each other, which means genetically identical clones, that can be a problem, being a clone, because if you are a clone, there's no diversity within a population, which means it's really easy to wipe out the population. Okay. Well, one of the things they find with bacteria is bacterial populations, there is genetic recombination. There is, like, these two should be exactly the same, and these two would become four that are exactly the same, which would become 8 and 16 and 32, and it amplifies pretty quickly. All of a sudden you get into millions of bacteria that should be genetically identical, but when they go through populations in nature, they find that there's a lot of differences. There's lots of chances for bacteria to get new combinations of genetics, which shouldn't happen with cloning. Okay. What should happen with cloning is every now and then, very rarely, you get a mutation that causes a different phenotype. Okay, and we're going to go on the next slide and look at an experiment that kind of displays how this genetic recombination, or why it's more than just mutations that are causing bacteria to change. So here we go with that. Okay, here's our experiment that we're looking at. We've got a couple strains of bacteria. Right here we have a strain of bacteria that has a mutation in it so that it is unable to make the enzyme, or excuse me, the amino acid tryptophan. What that means is that in order for this to live, it would have to be in some sort of medium that has supplemental tryptophan in it. If we were to put it in something that doesn't have tryptophan in it, it would not live. Okay. The other type of bacteria that we have here is one that has the tryptophan gene. It can make that itself, but it cannot make arginine, another amino acid, which just means it would not, if it was not given arginine, it would not be able to live. So, as would be expected, we're going to take some petri dishes down here that have what they call minimum medium in it, and the minimal medium basically means that it doesn't have any extra amino acids in it. Okay, so it's got some sugars and some things like that. But if these bacteria that grow in it, if they're going to need amino acids, it's going to have to make them itself. So what would be expected is if I take these bacteria and put them on this plate, they won't be able to grow because they won't be able to make the tryptophan that they need. Okay, and that's what happens. They don't grow on this plate, so I'm going to put a big X through there. Same thing over here, the ones that were missing the arginine gene, if I put them in this plate, minimal, medium, they won't be able to live because they aren't able to make the arginine amino acid that they need for protein production. Okay, what they find is if they take the two of these, this strain that's missing the arginine gene, this one that's missing the tryptophan gene, if they combine them, okay, into a test tube and let them exist for a period of time, and then they pour this onto minimal medium, what you would expect is that all of the blue bacteria, as they're listed here, the blue bacteria that are in this, all of them would not be able to live on the minimal medium because they can't make their arginine. All the red ones that are in here, they should not be able to live on the minimal medium because they can't make the tryptophan. What they find is when they combine these, let them sit for a little while, and put them into the minimal medium, all of a sudden there's a few that pop up that can actually live. Okay, and I say a few here, they're showing 6 different bacterial colonies, which would mean all the bacteria that were in here, only 6 of them ended up being able to live. But what that means is that 6 is a number that's a lot higher than what you would expect from just random mutations and replication. So what it means is somehow the tryptophan gene from this and the arginine gene from this, somehow they're combining into the same bacteria. This is giving us an idea that somehow bacteria, they can exchange genetic material, or they can join genetic material, or something like that. Okay, which is huge for them because it means that now we've got a bacterial strain here that genetically did not exist before. We've got genetic recombination. In eukaryotes, that's common. Every single time that we make a new organism with eukaryotes, we have meiosis first, which cuts the number of chromosomes in half, and then we have fertilization, where you're taking bunches of DNA from one parent, bunches of DNA from another parent, combining them, you have genetic recombination. You have a new combination of genes. That's good. With the new combination of genes, that means that there's a lot of diversity in eukaryotes, and all of a sudden it looks like somehow we can get that genetic recombination happening in bacteria, which is hugely to their advantage. Okay, as this chapter goes on, or as this section of the chapter goes on, we're going to find that there's 3 different ways that this commonly happens, this recombination of genes from bacteria, 3 common ways it happens. We're going to detail them. I think it might take probably another lesson after this one, but there has been a ton of study done on this genetic recombination as it has in bacteria. So here we go with this. Okay, the first way that bacteria can go through genetic recombination, again, that means genetic information going from one bacteria to another bacteria, or new genetic information entering a bacteria. Our first way is transformation, which was actually mentioned earlier in the year, transformation here. And so I have a diagram from chapter 16, if you can remember this experiment. This is the Griffith experiment, when they were figuring out that DNA is actually what causes genetics, okay, or heredity is caused by DNA. Anyway, if you remember this, these are different types of pneumonia bacteria, Streptococcus pneumoniae, and there was 2 different types. There was a smooth type, and if you had a smooth strain, it would kill you. If you had a rough strain, this is the R strain, it would not kill you. Or we're looking at mice here, not people. But anyway, so you take living smooth cells, put them in a mouse, the mouse dies. Take living rough ones, put them in a mouse, the mouse is healthy. If you take killed, so these are smooth strain, but we've killed them by heat, you put them in a mouse, the mouse lives because the bacteria is dead. So this is the crazy one. If you take the rough ones that don't kill you, and the heat-killed bacteria that don't kill the mice, if I combine them, inject both of them into the mouse, all of a sudden the mouse dies, and when we do samples from the mouse, we find living, smooth bacteria in here. Okay, and so the question becomes, how in the world did you get living, smooth cells from a combination of dead smooth and living rough cells? Okay, and so the idea came about this term transformation, and the idea was that Griffith put together was that somehow information, something, is getting from these dead smooth ones into the rough cells. Okay, they call that transformation. What ends up, we understand the process a little bit better now, and so I'm going to delete these things here. Just do a drawing for this. We understand these things a little bit better. But essentially what happens is you can take a living bacterial cell, and it has its DNA in here, okay, and then you can take another bacterial cell and kill it. And when I kill this thing, oftentimes when you kill cells, they just kind of fall apart, or lyse or break or whatever you want to call it, but they fall apart. And so this, if I kill it, I'm going to get little chunks of stuff everywhere, including chunks of DNA. So we're just going to squiggle these. Some of these things are chunks of DNA. And what they find out is transformation. Some cells are really good at this. Some bacterial cells are good at it. Some of them are not very good at it. But there are some cells that are designed with little recognition proteins on the surface. So if I look on this, it would have little recognition proteins on the surface, so that if they are ever exposed to DNA from the same type of cell as themselves. Okay, so let's say this is Streptococcus pneumoniae, and this is the smooth version, this is the rough version. They're both Streptococcus pneumonia. The only difference is a gene, a specific gene for that capsule on the outside. Okay, anyway, so let's just say that this is DNA from the smooth strain. Well, our bacteria here can recognize that this is DNA from a similar bacteria, and it'll actually take it in. So I'm just going to, in a rough way, just draw this. It'll actually create some pores to allow the DNA in. Once this DNA is in here, we can have, there's ways for it to basically, ways similar to crossing over, where it can end up inserting itself into the genome here, okay, where it's going to find similar sections of DNA and kind of swap in here. Again, similar to crossing over that happens in eukaryotes. But basically what we've done is we've got a genetic recombination happening here, where DNA has entered the cell. Okay. My hope is in class in the next week here, week and a half, that we are going to be able to do a bacterial transformation, and what we're going to do is take some E. coli, and we're going to put it in the presence of chunks of E. coli DNA, or sorry, not E. coli DNA, but chunks of DNA that contain the gene for glowing. Okay, the bioluminescent gene for glowing, and that glowing gene shows up in lightning bugs. It shows up in certain types of ocean organisms that live deep in the ocean, and they can kind of produce their own light kind of situation, but it's a glowing gene, and we're going to take that genetic code and we're going to try to get the E. coli to take in that code for the glowing gene, which means we will ultimately be able to make bacteria that glow, okay, glowing bacteria. The way you have to do it with E. coli, they aren't very good at opening up pores for DNA to come in, and so what you have to do with E. coli is put them in a specific scenario with some heat and some calcium ions, and basically make them uncomfortable to a point where it will open up some pores for the DNA to be able to go into the cell, and then hopefully become a part of the genome of the cell, so that it'll start making these glowing proteins, and we'll have glowing bacteria. But that's transformation. That's as deep as we're going to get in with that. But again, just the idea that it's taking genetic information from another cell, inserting it into a new cell, and now we have a combination of genes that we didn't have before. We've got, in this case, we've got the smooth gene entering into a rough bacteria cell, and we have transformation, okay, or genetic recombination. I'm going to end this video right there, and we're going to do another video for the rest of section 18.3 to discuss the other types of genetic recombination in bacteria. So enjoy.